In 1952, Alfred Hershey and Martha Chase conducted a series of experiments to prove that DNA is the genetic material of a bacteriophage, or a virus that attacks bacteria. The virus particles used for this experiment were simple bacteriophages consisting of an outer structure made of protein and an inner core consisting of DNA, as shown in the diagram. Hershey and Chase were aware that the phages attached to the host cell surface and injected some genetic material, either DNA or protein, into the host. Before conducting the experiment, Hershey believed the genetic material would be protein. To test their hypothesis on whether the phage was injecting DNA or protein into the host bacteria, Hershey and Chase set up two different batches of phage. One labeled with a radioactive isotope of sulfur, found in proteins but not in DNA, and one labeled with a radioactive isotope of phosphorus, which is only found in DNA, not proteins. Each phage was then used to infect a different culture of bacteria and was centrifuged to separate bacteria from phage parts and debris. Since bacteria is a heavier material, it will sink to the bottom of the test tube as a pellet after centrifugation, and the lighter growth medium and phage debris will remain at the top in what is called the supernatant. When Hershey and Chase examined their results, they concluded that a significant amount of the labeled phosphorus appeared in the pellet, while the sulfur remained in the supernatant with the phage protein. This allowed them to conclude that DNA was injected into the host cells and made up the genetic material of the phage. I will include a helpful link in the description below if you would like to know more about Hershey and Chase's experiment.